from Sean Burke of DeNovo Software, moving from data to results with FCS Express, Spectral Unmixing, and beyond.
All right, hi everybody. Thanks for hanging on the call here for a few hours. It's really kind of cool to see that not only do we have folks from the New York area, there's folks from all over the country. And again, many thanks to Tina and everybody at MetroFlow for continuing on to put all this together. It's been a great day to listen to some of these talks already for me. So I'm going to kind of pick up on the spectral data analysis a little bit here, kind of talking about working in this in third-party softwares like FCS Express, but we're also going to talk about the context of, not just getting kind of basic plots and statistics, but what FCS Express can do, and that's a little bit of the and beyond in the title of this talk here. But one place I like to start, I know, not everybody is using FCS Express yet, although a large number of people are, is kind of why would you even want to use it to get started? And what I like to do is I like to make this akin to technology convergence. So in the past, essentially what's been going on, or what was going on, is you needed a number of different devices to do a lot of the kind of standard things you do in your life these days. So you needed a GPS to get around, a camera to do certain things. You may have had this actual Nokia phone. I think I had that one. But all of these technologies, especially this little Nokia, they did one thing, right? It made a phone call. You could send a $1 text message, maybe play a game of Snake. So maybe they were multi-use. But all of this stuff is wrapped up now into your smartphone, and that makes life very, very convenient. And the same thing can kind of be said for flow cytometry data analysis. Essentially, what people are doing is they're using, not FCS Express, but if they're using other cytometry software packages, essentially they're having to take that data and copy and paste and go out to Excel. If they need to get it ready for a presentation, they need to kind of copy and paste and move things out to PowerPoint. And I know a lot of us have been guilty of kind of wasting time and putting little white text boxes over access labels in PowerPoint. And then even if you need to go out and use tools like Prism for advanced statistical analysis, well, all of this is wrapped up now in FCS Express. So imagine moving a gate in your software, and all of your downstream pie charts and bar charts and standard deviations and p-values update. And that's what we're going to kind of show you today. The other thing that comes into this is also spectral unmixing. So a lot of the unmixing that you can do in either Sony software or the Cytec instruments, as long as you have those reference controls, we can perform a lot of that in FCS Express as well. So to kind of recap, the big difference between FCS Express and other software is in other softwares, you're just kind of getting these basic plots and statistics, which you have to copy and paste over and over again out to other softwares before you get a final result. And really what we're going to achieve in FCS Express is you're going to get that final result directly in the software. I'm going to show you this through a series of a few examples, and then we're also going to show you how to get a final result out of a spectral cytometry experiment as well. And I'm going to walk you through how the new spectral mixing tools in FCS Express work. So essentially, there's a number of different results that you could have. Some of the examples we like to show people are for lab presentations, comparisons between samples, kind of gathering statistical information and statistical tests. And again, what you end up with other software packages is you need to, again, move copy and paste. You generally end up with lower resolution graphics. So if the publisher calls for 700 DPI, you have to use Illustrator to do something. If you have to calculate p-values and a ANOVA test, you have to use Prism. And again, this takes a lot of time and introduces needless complexity into kind of our basic data analysis. So I'm not going to go through all of these examples today, because I do want to get to the unmixing. I think that's a really cool new feature in the software. But I do want to show you just kind of one quick example of some of the graphing functionality in FCS Express, so you get an idea of how this works. Now, I just opened up FCS Express, and you may be thinking, oh, this looks like PowerPoint. This is not. This is FCS Express, but the whole idea behind the software is that shamelessly by design, we model everything off of PowerPoint and Office, because we know everybody on this call, everybody who's working in a lab has used PowerPoint at some point in their life. So, the same things, moving, resizing, grouping, all that kind of stuff is here in FCS Express, but this is your actual flow data. So if I click on an axis, you can see all the different parameters. But the really cool thing here is that if I move a gate, and you're going to see this, all of your results, bar plots, scatter plots, pie charts, all of this information updates in real time. And it's all because of the way that we link up to what we call spreadsheets, or what are spreadsheets in the software. You can kind of see this down here. And these integrated spreadsheets make it as easy as working in Excel, but working with your data as you move gates in real time. So it gives some really powerful functionality. I know when I used to work in the lab, oftentimes my PI would come back and say, all right, well, move a gate, just a pixel, and see how that changes all of our downstream graphs and all the stuff in Prism. And then we go and look at those graphs and decide, all right, well, that pixel changed and it make a difference, go back and undo it. And then, a whole hour had gone by. But again, what we can do here is just move that gate, see how that makes a difference. And I kind of like this little scatter plot. It looks like a happy face, right? It looks like we're pretty happy there. And if you kind of move this around, I think we can make it sad too at some point, right? So that's not where we want our gate. But essentially you get those updates in real time, which makes this a very, very powerful tool to either work as an intermediary between going out to Excel and Prism, or simply to kind of replace that altogether. So again, that's kind of what we mean in FCS Express by getting results. Getting those bar charts, getting those pie charts, that type of thing is a result. And in FCS Express, we have fully integrated analysis and presentation tools. All this stuff is going to go out at high resolution. The integrated graphing and statistical analysis is going to save a lot of time. And there's no need to copy and paste. I know a lot of people are stuck copying and pasting out to Excel. If you do that 100 times, you're going to end up making a mistake, right? So it's just going to reduce human error to a certain degree. And again, this is going to bring down time, cost, and complexity when it comes to putting together your data analysis. Now, in FCS Express, we know that there's also a lot of specialized analysis that people perform. So we're really well optimized for working with things like high content data, really big data sets, working with any kind of transformation tools, K-means, TISNE, VISNE. There's actually more of those coming in a release of the software in just a month or two. But one of the things I wanted to focus on today is also spectral data analysis and unmixing, because this is something we've essentially have worked with Sony, we've worked with SciTech in the past to not only read the standard FCS files that come off of these instruments, but actually to read that full spectral data. And what that means in FCS Express with that access to that full spectral data is you're not just limited to looking at, CD4 versus CD8. You can look at the full spectrum of every single event in that data file. So you can use the standard FCS files, you can use the full kind of spectrum that's in there, you can use them both interchangeably, and you can do that quite simply, which you're going to see here in a minute. But also, just as of about a month ago, we released a version of FCS Express, or a new update that enables spectral unmixing. And I'm going to show you that today. I'm going to show you how easy it is to do. We can do it live on a call here because it's pretty simple and straightforward. And by integrating the spectrum plots with spectral unmixing and all of the other tools that we have in the software, this makes analysis kind of even more efficient and even easier to get that result that you're going for. And what you're going to see is, again, you can use these analysis layouts over and over again, so you don't have to recreate the wheel every time you're performing an analysis. So I want to show you what it kind of looks like here. Let me get out of my presentation mode there, and we'll come back over to FCS Express. So this is an example of some spectral data that was acquired on a SciTech instrument. What you'll notice when we load this data into FCS Express, we've just loaded in the standard FCS file. But if you're loading in data from the Aurora instrument, if you're loading in data from the Sony instruments, as long as you have the set of data that includes the raw spectral information, I believe in the Sony instrument, they export an SRAW file. In the SciTech instrument, it's just about where they store files. So as long as you keep that information in the same place, essentially what you'll be able to do is come in here to the Insert tab, and you'll be able to insert one of our spectrum plots. And when we insert a spectrum plot, you're going to see that we're going to be able to access the full spectral information for this data set. And again, Jeff did a lot of the kind of heavy lifting for me here to explain what these plots mean and how they work and how to acquire them. But essentially, we'll gather all of that information, and you can work in FCS Express just like normal. So if I have a gate on my lymphocytes population, I want to apply that gate to my spectral plot and look at the spectrum for the lymphocyte population, I can do so. And additionally, what we'll get is as we move gates around in FCS Express, the information that's being displayed on that spectral plot is going to update for us in real time. So if I move that gate, it was called lymphocytes, up around the monos and the granulocytes, I can see in real time how that spectral information is changing and how it's updating and how it's applied to the different populations in the data set. So it's a very powerful tool being able to kind of visualize this information in real time. You acquired all of this spectral data, so it's a shame if you can only export the FCS file and only be able to work with that. But again, with any of the spectral instruments that are on the market, FCS Express is going to be able to read that data and pull it in. And of course, there's a lot of different ways to display this. If you don't want to look at the full density plot and you want to look at kind of individual spectrum from different populations, here's an overlay of my CD3 positive and my CD3 negative. Well, again, you can create those overlays very quickly and simply in the software. And again, you get that real-time updating as you move a gate to kind of compare what these spectral signatures look like between different populations. So again, the rest of the analysis that you perform in FCS Express works just like it would for any old FCS file, except that with any of the spectral data, you can go back and you can generate the spectral plots, you can format these. And again, all of this is easily reportable. So if I wanted to take all of this information, including my beautiful looking spectrum plots, and take it out to PowerPoint, we can export all of this directly to PowerPoint. And what FCS Express is doing here, it's saving this out and it's going to launch up PowerPoint. So we're back in PowerPoint now, we're not in FCS Express. But the really cool thing is that you get these high resolution, what we call vector graphics. Vector graphics can be moved, they can be resized. It's not just one big continuous low resolution image. And the other kind of nice thing about these exports from FCS Express is you can even ungroup these things in PowerPoint. So if you don't like CD15 on the axis and you want to call it, CD15 is awesome, you can actually make the change directly, in this PowerPoint export. You could do it in FCS Express as well. But again, it just facilitates getting that end result very quickly and easily. So again, I want to kind of shift gears away from kind of some of the standard stuff that folks are used to doing in FCS Express to talking about spectral unmixing a little bit. And again, I have to say, thanks to Jeff for giving us a really great overview about essentially how spectral unmixing works in the previous talk. It takes a little bit of that load off of me, and I wasn't planning on spending a ton of time on that today. But essentially, when you're doing spectral unmixing, the problem that you have, well, it's not a problem, but you collect a continuous high resolution optical spectrum for every event in a sample. Kind of looks like this little gray box here. And the spectrum is the sum of all of the dyes present in the cell or the event of interest. And again, spectral mixing is the process of transforming that data so that we can look at a plot like this, where we're showing that we have some yellow dye, some green dye, some purple dye, and some blue, which is kind of unstained events that make up this observed spectrum, and break that down into the pure components. And if we want to unmix this data, essentially there's a series of mathematical calculations that can be done. What we do in FCS Express is use ordinary least squares. I know Jeff had mentioned that they're using weighted least squares in the Sony software. What we're working on doing in FCS Express right now is we have ordinary least squares. We're going to be adding additional unmixing formulas and methods in some new updates to the software that you'll see coming. So you'll have the ability to work with multiple of these and kind of compare them side by side. But essentially, just kind of as Jeff mentioned, provided a series of reference spectrum, which is the A in this formula, and we have our measured spectrum, which is Y in this formula. We can do some sort of mathematical operation to arrive at C, which is going to be our spectral unmixing matrix, more or less. Again, in the interest of time today, I'm not going to go over any of the more details of this, but I want to show you how easy it is to perform spectral unmixing in FCS Express. So essentially what we do is we go into our Tools tab, and when you update FCS Express, you're going to see compensation and unmixing, and we're going to be able to come in here and choose unmixing. So this works just like you would perform compensation in FCS Express. There's a series of steps that you follow. Step one is add some single sane controls. So I'm going to click on this to get that launched up. And I'm going to go in and look at my reference group or my single sane controls. I'm going to exclude my beads population here today. I have a CD123 cells and beads. Just for the sake of simplicity, I'm just going to work with the cells today. But this is really the only kind of time hit you're going to take in FCS Express. Right now, I'm just waiting for the data files to load into the compensation or into the unmixing tool here. What FCS Express is also doing in the background, it's reading the data file names, it's getting ready to prepare labels for you automatically. It's trying to determine whether your cells or your sample represents an unstained control, whether it represents some sort of single stain sample. So it's doing all that as it's pulling this in. But as we perform the rest of the mixing in here and we get that applied to a plot, everything is going to update in real time. And here comes all of my data. And as a quick overview to what's going on here, essentially we have all of our data files that are loaded into our little wizard here. We've automatically labeled them, so we try to pull the label from the name, but if you wanted to label these, anything differently, you can, put in any sort of information that you want there. Oops, and I hit undo, so let me redo to get that back in. And I apologize, it's gonna take me a little hit here to load all my data files back in. But essentially we can label those with anything that you want. And what you're going to see once this data comes back in is that in the sample type, I'm going to be able to define whether this is a control, whether it's an autofluorescence control, whether it's a sample. And then in our negative control section, we're going to be able to assign a specific file as a negative control. If we're working with an autofluorescent control, we're going to be able to say no negative control because we won't need one for that. And when we pull in here, again, we can say self-negative, right? So there's a few different options, and really just about every single type of situation you could run into with your controls is going to be applicable here. So I'm going to kind of skip through some of this stuff. I'm going to say we're going to calculate the matrix for the full spectrum of data. I'm going to click this build histograms button. And essentially what FCS Express is going to do is it's going to build out a series of pages on the bottom here. You're going to see these popping up in just a second. And it's going to contain one page for every single sane control that we've put together. And I can kind of move this box out of the way so we can take a look at it. And what I'm going to do is that for each page, I'm going to go through and make sure that my controls make sense. So in this case, where we started here is we have a scatter gate for the CD15 control, but I do need to change this gate because there actually isn't any CD15 positivity in that particular population. But you can see that as I change that gate around, when we identify the positive population, the histogram and the marker is going to automatically update and try and find that peak, and we'll arrive at that spectral signature that we need. So again, what you're going to do is you're going to go through, all of the pages that are here. And again, this is kind of like, performing compensation in FCS Express. The same thing applies. You have to go through and, just make sure that the peak fitting did its job well. In most cases, and since the release of FCS Express 7, The peak finding algorithms have gotten better and better and better in the software. But again, if you end up in a case like this, you need to move your gate around to a different scatter population for that. You can do so. And again, here's that CD123. So I mentioned I'm not using beads for this one. If I wanted to pull in the beads control, I could, but I do know I have a little tail of the population up here, which is actually giving me the spectral signature I need for that. And again, just kind of working through the rest of my controls here, out to CD4 and into my unstained. And again, once I'm happy with how all of the controls are set up, I know I just kind of rushed through that in the interest of time. I'm going to come down here and I'm going to click Calculate Matrices. And you're going to see FCS Express calculates that matrix pretty darn fast. It just popped right up. And what you're going to see is that for every wavelength or every detector that we have in the system, we're calculating a new parameter for that, which is the unmix, which is going to be the unmixed parameter. We're calculating that across all of these things. And if you had autofluorescence parameters, you'd have autofluorescence columns in here. The other kind of nice thing is you can turn these on or off if you're using them or not using them that day. They kind of go with the idea of a spectral library. Now, to get this applied to some data, again, when you're doing compensation in FCS Express, the same kind of rules apply to unmixing. You probably want to use your unmixing layout as a place to do your unmixing and create a new layout to actually perform your analysis. But again, just for the sake of time today, I'm going to load up a data file into my data list here. I'm going to pull that into FCS Express. And by default, we're not going to apply any unmixing to this. We're just going to insert the plot. I'll get that in here. So what you'll end up seeing in this plot by default is just the scatter parameters. We don't have any unmixing applied to this. But what we would do is you'd drag and drop that unmixing to this. You can see that now we have all of our unmixed parameters. And just like compensation, you can assign a default. If I say right click on this unmixing and say set it as the default, anytime I load any sort of spectral data into FCS Express, we'll use this unmixing matrix automatically. So that way you don't have to drag and drop, you don't have to apply. But again, this just works like standard flow data now. You can create a gate. So if I create some sort of gate on this population, we'll connect that gate, and my gate dialog window is somewhere buried underneath all my Zoom windows. So let me create a little gate there. And if we drag and drop and apply that out, we can start using any of the unmixed parameters. So if I want to look at, say, CD3, versus CD4, right? We can see how this data was unmixed. We can look at it. And again, if we need to go back and make any sort of change to the unmixing, we can just go back over to our little unmixing navigator here. We can change the gates. We can do anything that we want to have that unmixing updated, and then that will apply to these data sets. So again, with FCS Express, the unmixing tools in here are very straightforward. They're very easy to use. Again, what you're going to see in the options category here and not too long from now is the ability to use ordinary least squares as well as other methods. And just like compensation, you can create as many unmixings as you want, right? You can see here I added another one here. And when I have additional algorithms to select, I can select a different algorithm, apply that over to the data, and I can compare, how these two algorithms are working side by side. And we know that's something a lot of people had asked for. And again, that kind of stuff is coming in the not so distant future for FCS Express. So what I kind of wanted to recap on a little bit today, again, I know we went through a lot of stuff here kind of quickly in the last 24, 25 minutes, but if you can remember, time taken for analysis, when you talk about all this stuff, it's actually a minor part of the process. What we found when we started talking to folks is that reporting data, creating graphs is the type of things that's taking them a long time. So getting things ready for publication, copying and pasting out to Prism and Excel, performing unmixing, performing compensation, getting those results can be difficult. But when you're using FCS Express, we make the time it takes to get those reports, get those graphs, a very small part of the process. The analysis portion, if you're using other software, this generally goes down in FCS Express as well, because a lot of the time savings and efficiency tools that we have in the software. So that kind of leaves us, right? How do you want to spend that time? In FCS Express, we want to see our customers and our researchers going back to the lab bench, publishing, putting together posters. Believe it or not, eventually we're all going to go back to going to conferences and doing these things in person. So we want to see you going back there as soon as you can, versus sitting in front of the computer and copying and pasting, putting little white text boxes over access labels. Again, these people are very frustrated at having to do that all the time because they're not using SCS Express. So with that, I just want to take, again, a few moments. We'll answer some questions. I wanted to say, again, a big thank you to all the folks involved with MetroFlow for putting together this kind of virtual meeting for us. It's really great, and I still see that we have over 100 people hanging on the call here for a few hours, which I think is pretty impressive. So many thanks for everybody coming out today. So with that, are there any specific questions that have come up that I can address?
Yes, thank you, Sean. That was absolutely wonderful. And we definitely have a couple of questions for you.
Great.
So the first question that came through on the Q&A from Yolanda Menke was, don't you need a minimum number of positive events to calculate a good matrix? There was one example that looked like it had very few positive events, but the impression might be because of the scaling, might also be to the scaling.
Yeah, I think you're referring to the one plot where it looked like there was a very tiny tail. I forget which marker it was on the histogram. I mean, essentially, you need to figure out the median of a population, and that one particular example, actually it was CD123, I remember. We do have a beads control for that as well, because the cells control wasn't the particularly best way to do that. So yeah, I mean, there are some minimum number of events. I guess there's nothing empirical that I can say you need 57 events at least to determine what the MFI value is, but really the same kind of rules that you would use for determining single sane controls for things like compensation can simply be applied to unmixing. I mean, you need to be able to figure out where your positives are and your negatives are accurately. As long as you can do that, that's the minimum number of events that you need to do that.
Okay. And then we had some questions from the chat, so I'm going to go back and forth. Rohit had asked, is the algorithm of spectral unmixing similar or dissimilar to Sony or SciTech? And then their follow-up question was for MFI, how is it calculated using only the highest emission channels, or the whole spectrum?
Yeah, so everybody's algorithms are a little different. This kind of stuff is generally proprietary. We're using the standard ordinary least squares method right now. As Jeff mentioned earlier, Sony's using a weighted least squares. And again, what you're going to see in the not so distant future is FCS Express is going to offer a number of different algorithms. So you can actually, right now you can pull in your data that's been unmixed in other software. You can redo the mixing in FCS Express, and you can compare side by side if you'd like. And again, in the not so distant future, you can use any of the algorithms that we use to kind of make those comparisons as well. Essentially, what we're using for the median or the MFI value in FCS Express, let me see if I can pull that up, is essentially the median for this marker, right? For whatever this marker is set. And what FCS Express does is it finds the channel. Here you can see this says 783. If I come over here and I look at CD16, it says 661. So it's looking at the MFI in that particular channel or that particular detector for the information that falls in that marker. So that's how FCS Express is pulling in that information. That's what's being used for the matrix. And if you're not happy with the pick that it makes here, there are some ways that you can go in. You can change which detector it's using. But again, that's meant for a little bit more of an in-depth conversation, or you can come back to our website and look at some of the webinars we put together for that. But that being said, any additional questions, please just reach out to us at support@DeNovoSoftware.com. You can also visit our website at DeNovoSoftware.com to get a free demo or to pull down the latest update that will allow you to do this sort of stuff.
And it actually looks like we're going to have a little bit of extra time because our last talk for the day did not pan out, it seems like. So we can actually take this time. Sean, there was an extra 2 or so questions if you have the ability to answer. And then if there are any remainder questions and the remainder of our speakers are still online, we might be able to address those as well. Are you okay with answering 1 or 2 more questions, Sean?
Yeah, absolutely.
Perfect. So one question also from Rohit, and then we have another one from Thomas. So Rohit asked, is there a better unmixing algorithm to analyze dim express markers?
Well, we only offer one unmixing algorithm, so that's your choice. you have one choice. We make the choice simple. So we're actively looking at other algorithms, and I think we'll be able to answer that question more accurately once we implement some of these other algorithms and we can do some comparisons side by side. So I'm not sure, but again, right now you have one, so you can check that out.
Okay, perfect.